Hi all, Dr. Clark here again. Today for Natural Resources, we're going to talk about environmental risk. All right, so we talked about environmental health, we talked about toxicology, and now we're going to talk about the science of environmental risk. Where's the science behind things like heavy metal bioaccumulation or toxic chemicals in the water system and things like that? How do scientists evaluate these systems? What are the regulations around some of these? And just kind of a brief overview of how scientists go about environmental risk estimates. Okay, so first of all, we're going to talk a little bit about bioaccumulation. So bioaccumulation often happens when things like non-polar toxins are stored in fat. So it can also happen with polar substances, but often it's non-polar substances get stored in fat tissue of different organisms. And it accumulates, from small organisms to large organisms, and the effects accumulate on that. So for example, let's say we have a toxin like DDT, okay, which used to be used to as an insecticide on fields. In the 50s, it was created in the 40s for World War II, but then after World War II, DDT was used for a good period because it's an excellent insecticide. It kills all the insects. But it can accumulate in the bottom of rivers and streams and lakes and things like that. Small insects can then pick it up. It really does nothing to the small insects after it's in the system. It does really nothing to them. Fish then eat those small insects. Fish, it depends on the fish species. Sometimes they'll have some effects to the fish species, but then the birds will eat the fish, and then you get this huge accumulation in the tissues. And this is exactly what was happening to fish-eating birds in the 60s when, books like Silent Spring, so Rachel Carson's work on, birds came out, when Jim Greer's work on bald eagles came out. It was due to, DDT. Their work was mainly DDT work, but it was because the bioaccumulation was happening at these higher trophic levels. So where you weren't seeing much science in the fish and the insects in the system, that's where we were looking. Okay, we knew the runoff was going out of the fields, so we'd check on the fish and the insects and stuff in these water systems, and there wasn't a huge effect on them. But we had to draw that connection between that and, say, bald eagle nesting. And what happens when DDT accumulates in the tissue, it gets into the bird's system, and then it reacts with the capability of the bird to lay down shell material on the eggs. And so it caused the eggshells to be really thin. And so the egg would come out very, very thin, and sometimes it'd come out and just shatter when it hits the nest. Okay, but other times the bird moves on the nest or might just barely touch it with their toe, and it would shatter the egg. And the eggs were so porous and so devoid of calcium that they were just falling apart. And that's when we saw huge declines in birds that eat fish, and the connection between that was drawn by Carson and Jim Greer and a bunch of other biologists in the 60s. So bioaccumulation is that toxins accumulating in the tissue. Okay, biomagnification is normally when those toxins accumulate and become a burden, more toxic to the individual as you move up trophic levels. Okay, and so you can get accumulation, okay, and biomagnification working together, like DDT, where you accumulate these concentrations, and eventually over time the toxicity builds up and then it becomes toxic. All right, persistence. So persistence is basically, like I talked about before, the half-life of a chemical, or how long does a chemical persist in the environment. Is it persisting in the tissues, like the fatty tissues of organisms? Is it persisting in the soil? Does it persist in plants? And how long is it stable in the environment? When we're creating chemicals for herbicides and pesticides and things like that, we want them to degrade very rapidly. Okay, we don't want them to bioaccumulate and biomagnify at all. And we already learned our lesson with DDT. Well, I hope. Um, but so most modern pesticides, they degrade very rapidly once they're in the system, once they're exposed to water and things like that. Now there are things though that we still constantly use, okay, that have persistence for a long time. Metals, whether that be lead or um, mercury is still used in some systems. So there are definitely metals that can leach into aquatic systems and persist for a long period of time. But I think the main system that we're interested in now are what we call BAPs or BPAs, and that's particulate matter that's normally coming off of plastics. And so whether that actually be physically the plastic or the chemicals that are laid to keep plastic from deteriorating and things like that, they can be toxic and they can be stored in the environment for a long period of time. I mean, for example, there's a lot of restaurants and a lot of places that are banning straws, and some of these straws have BPAs, and so they do persist in the environment for a long period of time. And it's really coming about from the photographs of tortoises and things like that got straws stuck in their nose and whatnot. But even more than that, the science suggests even once the straw breaks down, you still have minute microscopic plastic particles all over in the environment that can bind up other organisms' bodies. So insects and things like that can digest these. They can't actually break them down, so they'll bring it into the system, but they can't get rid of it. Sometimes they binds up, and you can find these BPAs in the system much longer. So other persistent organic pollutants that we've seen issues with, things like flame retardants, whether that be clothing or just materials in walls or coatings that go on to different things. We know that they can be extremely harmful for the nervous system, and now we're seeing it all over the world that BPDEs are persistent, and we use them mainly in flame retardants to prevent fire. Other things that we now know it was a good idea when we had it, non-stick plastic coatings that we see in like pans and things like that. They can definitely cause liver damage and are known carcinogens. They can persist in your blood for a long period of time, in the environment, in the water system for a long period of time. And so you'll start to see that these non-stick plastic coatings, at least certain types of them, are no longer used. But like I said before, it really takes this, we utilize these. It was a good idea, different types of flame retardants and the different plastic coatings and things were good ideas. They made people's lives much more advantageous to have these things around, but it takes a few years for us to find, whoa, hold on a second, when they break down, just a little bit even, they're a huge problem, and they can really be a health pollutant. Phthalates, these are, again, found in plastics, and they can mimic our estrogen, and they can increase, estrogen in males, which can cause them to, be more female-like. We've seen this a lot in animals like frogs and things like that, but there is some evidence that suggests that it has a direct effect on humans too. So there's lots of ways that these things work, and one way that we're more interested in when we talk about the science of toxicology or the science of environmental health really comes with chemical interaction. This is kind of the new kind of process to wrap our minds and wrap our hands around these ideas of chemicals that we're using constantly every day to preserve things, to store things, to make things smoother, so coatings and things like that. Well, what happens to them when they break down? And so you can have different types of reactions, like antagonist reactions, where one material will interfere and affect another material. So you might have a material that causes another material to break down when they come into contact with each other. You can have additive reactions where two chemicals that normally aren't, they're normally not together, but when they come together, they have an additive effect, which can be toxic. And then you can have synergetic reactions, things like, asbestos and smoking. So, asbestos exposure increased the risk of lung cancer by 20%, or 20 times, sorry. And smoking is 20 times also. But when you combined someone who works with removal of asbestos and they're exposing themselves to that, and they smoke, they're about 400 times the risk of a normal individual for developing lung cancer. It's almost a guarantee, pretty much, that you're going to develop lung cancer if you're a smoker and you work with asbestos. These are synergistic reactions where they're multiplied, so they multiply the toxicity of whatever it is. And so, this example is well documented, and all asbestos workers should be getting the document that says, you are at risk of developing lung cancer if you work with asbestos. If you smoke, your risk is now this. That's a pretty well documented piece of information. So we often want to introduce ways at which we can minimize the toxic effect. We know that we're exposing ourselves to toxicities constantly, every single day. I mean, from a candy bar with a plastic wrapper on it, how much of this exposure from just being in a plastic wrapper is an issue, or the drinking water that you're getting out of a drinking fountain or out of your tap. What kind of exposure are you exposing yourself to? Is it better to drink bottled water, which has BPA's in it, or has the potential to have BPA's in it? It depends. It depends on the individual, and so we often are interested in, minimizing the toxic effects of things. Okay? And like I said before, every material can be toxic or poisonous under certain conditions. You can actually poison yourself with water. You could drink enough water that you could kill yourself. You could drown yourself in water. So even the most beneficial product probably on the planet, water, can be poisonous at certain concentrations. Now, the issue often is, can we keep these toxins under a certain threshold? Right? And so we need to set thresholds, and that's what scientific environmental health agencies, organizations like the EPA, they set certain thresholds based on laboratory research on mice and rats and things like that, and frogs and other things. They set these scientific thresholds, and we need to be under those. Right? Sometimes it's wrong. Right? I'm not gonna sugarcoat it in any way. Sometimes that threshold is not correct. Mice are a good model for humans, but they're not a perfect fit for us. And so when we concentrate on doing these laboratory studies to look at toxic effects, we're doing it with laboratory animals, not on humans. And humans have different metabolisms. They have different exposures to different things. And we're often also not figuring out, well, what's the cumulative effect? So maybe we're after maybe it's a new plastic coating, a new polymer that we've developed, and we're looking at the toxic effects. Well, what happens if you scrape a little of that off and you mix it with this other polymer? Right? We often don't do those kind of experiments. Right? We don't do the additive experiments, which can be really concerning, because that's what's going to happen in real life. You're not just going to be exposed to one chemical, one toxin. You're going to be exposed to many toxins at the same time often, and through many different sources of material. So, we can look at metabolic degradation, and this is often how we do these kind of studies. We take mammals, typically mice or rats, and we expose them to different toxins, and then we look at how degraded or how much degradation their liver has had over a certain period of time. We also try, in some cases, to reverse the process. So is there ways at which you can reverse it? So if you supply and apply this toxin to the individual, and then you remove that toxin. So a good example is like Flint, Michigan. So the exposure to lead in the water system, these kids are all exposed to it, not knowing the danger of the system. They're exposed to it. Now, let's say the water is clean. I'm not saying the water is clean yet, okay? Because there's reports that say it's not clean. But let's say the water gets cleaned up, and the kid, is now, it was 2 years old, and now they're 6 years old. 4 years later, we finally get the water system cleaned up. Is that individual going to recover over their lifetime? Or do you get these degradation that happen for those 4 years, or really for the exposure of 1 year? Was that enough to cause a toxic effect, a detrimental effect to the individual for the rest of their life? We try to do that in the laboratory with mice. And we're often interested in those products where, like I said before, you have this product, it's a great product, maybe it's a new product, and it's been tested, the product itself's been tested many times, and it passes with flying colors. Great, it's good stuff. Right? But then it breaks down over a long period of time, or over some other chemical being exposed to it. It breaks down, and now it produces a harmful chemical or a toxic chemical. And those are ones that we're really interested in. Excretion and repair. And like I said before, if these are hydrophilic toxins, a lot of times they can be excreted through the urine of an individual. This can be helpful, but this can also be detrimental depending on where that toxin is being excreted into. If it's excreted into a sanitary system, like we have in developed countries, that's probably good. Okay, that water is going to be treated before it's dumped into a river. If it's excreted into a non-sanitary form, so it's just excrement goes directly into the water source and then it's drank again, this can be a huge issue. Or if it's excrement from wild or from domesticated animals, wild animals are going to drink that up. There's lots of situations where the environmental toxins can be excreted but then picked up and cause even more problems. You can also excrete things by breathing. So you excrete carbon dioxide, but you can excrete some of the chemicals that you would be picking up from air, and you can excrete it right back out. Now, I talked about this a little bit before when I talked about mutagens. Okay, so we have a DNA repair system that's called P53 or protein 53. And that is a repair system that looks at your DNA and looks for mistakes in your DNA. It looks for thymine dimers or just wrong base pairings. And then it'll turn on a repair system, repair enzymes to come in, cut that out, and put new stuff in. So we have these built-in repair systems. Sometimes, though, the exposure to different things, different toxins and different carcinogens and things like that, can affect the repair system. So if you break the repair system, then there is no repair going on, and you can develop cancer and lead to cancer. And so that's often what we're really concerned with when it comes to carcinogens, is what's the chance that the carcinogen will affect the repair system and prevent any repairing of mutated cells or mutated DNA for the future of that organism. So over a long period of time, that increases the chances that the individual would develop cancerous cells. So how do we measure toxicity? Like I said before, first off, we do a lot of animal testing. Whether this be good, bad, indifferent for individuals. Now realize that animal testing is done on animals that are bred for the animal testing. So we breed mice, we breed rats, we breed frogs, insects, different things for the testing purpose. Now I know if you're of certain religions or certain beliefs, that all organisms are sacred, and that no matter what, we shouldn't be killing organisms for the benefit of anything, except for maybe for nutritious benefits to keep another organism alive. But for testing of chemicals and things like that, or for scientific testing, people have issues with this problem. Unfortunately, right now, this is the method that we use. In the future, it will not be the method that we use. Currently, there is a lot of research in creating synthetic tissues and creating synthetic organs. So you're just really recreating the genetics, and you're creating the synthetic thing. So no organism actually has to die. You just create the synthetic tissue in a petri dish or in a test tube, and then you're testing the toxicity against that. We have those capabilities. We currently do some of that, but it's not to the measure of animal testing that's going on. So we'd have to really be able to create an entire system and basically create life from non-living things, which is possible. We've done it before. We've created life from non-living cells, or living cells from non-living stuff. But we're a little bit off from creating an entire organism and being able to do this in a laboratory. So animal testing still happens, and it's a concern. It really is a humanitarian concern. I'm concerned when I see it, and we have thresholds of how many organisms have to be used and how many organisms can be used. So there's kind of a maximum number of organisms that should be used, and a minimum number, so we know whether or not the testing is legitimate. There's lots of situations. Like I said before, different individuals are going to have different sensitivities to different toxins. I mean, just look at people that you know and associate with every day. You can give them, a little bit of grass, and some of them are going to start sneezing. And you give another friend, a vial with dog hair in it, and they'll start sneezing. And just in the immune response to these benign things that we have in nature, so many people sensitivities are very, very different. that would also go for toxic things that we'll see in the environment. So for some things, toxicity is a piece of cake. I'll tell you, for animal testing, this is a problem. The animals that are used are almost always genetically identical. So there is no diversity of genes, and because they're all bred, with the purpose of, hey, we're going to use these animals, and then they're going to be cold off, they're going to die after the experiment. So they're all genetically identical, but that does no good if we have different sensitivities from individuals that might be exposed to this chemical. And so that's a huge problem when it comes to animal testing. Now, there is complications in measuring toxicity, but we often use what we call an LD50. Okay, so when we're doing an animal test, an LD50 means we're interested in when or what dosage does 50% of the population die at. Okay, so you use a certain dosage of a toxic chemical, and when 50% of the animals dies, that is the LD50. Okay? Now, the LD50 is not where we are like, all right, this is safe enough for humans. Okay, we don't want 50% of the humans to die, but we're after the LD50, so then we can calculate from there what's an acceptable toxicity. Okay? No humans are going to die. That's an acceptable toxicity. At least for most chemicals, we don't want the individuals to die. However, there are some chemicals that we have out there that we utilize that we know that the LD50 is, higher. And so it's kind of a give and take, because the more potent a chemical is, so maybe we're talking about antibiotics, and we're creating a new antibiotic, we want that antibiotic to destroy the bacteria that we're interested in. So we'll look at an LD50 of that antibiotic. And so the more potent it is, though, the more bacteria, the faster it will clear someone of a bacterial infection, but also the chances that you might have complications with that. And so we're often interested in LD50s. And again, like I said before, if this is done on mice, if it's done on rats, if it's done in frogs, insects, there's very different physiology, very different metabolism, plus you have those added effects. So an LD50 on a mouse is not the same as an LD50 on a human, but there is no way, and we don't want to see this, at least I don't want to see this, I don't want to see an LD50 done on humans where 50% of the humans are going to die due to that medical test. Most people are not interested in those kind of tests. Okay, so in the end, measuring toxicity of new chemicals that are coming out, new products, new preservatives, new food types, all these things can be very difficult to estimate the risk to humans. That's why I said before, often what occurs is we produce these products, they're seemingly safe. 5, 10, 15 years later, after they've degraded, we now find out, okay, they're not safe anymore. These are awful products, and they cause huge complications once they start combining with other things, or once they start degrading to a certain point. So toxicity ratings are based on a certain number of grams per kilogram of body weight. Okay? And so we have a certain toxicity rating, and it's based, basically it's just based on, if you have a 180-pound individual, okay, we're going to convert that to kilograms, and then how many grams can that individual have to produce a lethal dose. That's the toxicity rating. That's where we say, okay, this is the lethal dose of this material. From there, we can classify them in different ranking modes. So very toxic materials is 10% of that amount. So 10% of whatever the lethal dose is a very toxic material. Okay? Extremely toxic material would require 1% of the amount. A super toxic chemical, often it doesn't even register on a percent. Okay? It's a microgram. You know, very, very little of this material is lethal. And so we'll often rank things based on moderate toxin, very toxic, extremely toxic, super toxic, and we'll rank them based on those and give kind of a toxicity rating that's based on, you know, percentage of, you know, the lethal dose. All right, so acute effects are normally exposed or single exposure, and you get health problems from it. So maybe drinking water with lead in it is enough to cause health exposure, and you only needed to do it one time. Okay? Chronic effects are often the result of long-lasting or repeat small dosage of things. You know, repeat small dosage of, like, you know, carcinogens from smoking cigarettes is a good example. But a chronic can also be just one big large dose that's going to have a lifetime of long-lasting exposure that can cause a lifetime of health problems. Typically, acute effects are immediate health problems. Like, you feel it today, maybe it lasts a lifetime, but it was due to a single exposure. Chronic effects could be that you're exposed to a large dose, and then over your entire life you feel the results from that, or small doses that build up and then eventually cause whatever the effect might be. The problem that we often have is acute effects we know really well, because we can take a mouse and we can give it an injection of a certain toxin, and we can look at its immediate health problems. Boom, next couple, 24 hours, 48 hours, 72 hours, whatever it might be, but we can see the exact problem. Chronic effects, though, is very, very difficult, because we can expose that individual to that chronic dosage or small repeated dosage over and over and over and over again throughout their entire life. And in some individuals, you'll never see some health problems. And in other individuals, it can look like an acute effect. Like, you've been exposing it to a small dosage of a certain chemical, and then on day 30, boom, now you see this effect. And so it's hard to decide, well, is that a chronic effect? Was it building up in their tissues? Or is it an acute effect because they're now exposed to something else in the environment? It's really difficult to study these kind of effects. The other thing is we know that we know this for all organisms, but as humans age, our cells deteriorate. Our DNA gets older. So when, you know, you might have been young and you might have been able to be exposed to UV radiation constantly and never produce a thymine dimer. But now that you're old and your DNA repair system is old, now all it takes is, you know, one sunburn, and now you produce thymine dimers, and now you've produced cancerous cells. Okay, and so it's kind of difficult to kind of study these chronic effects. Also, you know, you can get very different dosage response curves. So, like, you know, you could take these different mice and we can put them in different groups, and, you know, we might say that the dosage could be, you know, you might get 20% response. So, you know, maybe this is death, okay, or maybe it's a behavioral response. You can say right at the beginning, any dosage, anything that's measurable, 20% of the population is going to respond to it. And then you're going to get, you know, an up curve from that, you know. Other things, other types of chemicals, it doesn't register at the beginning, and it's uniform, okay? Straight up. And then there's other dosages that might not register until you hit a certain threshold, okay? And then you see that upturn. Okay? So different different chemicals, different toxins, these kind of things can have very different response curves. Right? Even if you're just changing the number of hydrocarbons in something, or you're just changing the bonding of certain things, whether it has a double bond or a single bond, very simple changes can cause a very different response curve. And so for all chemicals, you kind of have to do these different response curves to figure out, well, okay, what's the baseline at? And you want to use genetically identical mice in order to do this, because you'll never get a baseline if you're using diverse mice. And so over time, you might have a baseline that's down here for one genetic strain, and a baseline that's up here for another genetic strain, and on and on and on. Now, that's what we really want to know, because if we're going to supply this to humans that are genetically diverse, we want to know what's the extreme. I mean, could you get a baseline clear at like 35% lethal versus 0% lethal? Is some people that are exposed to this, are they all going to die if they have a certain genetic disposition or a certain genetic code? Is it going to cause them to develop cancer if they're exposed to it, even the most minute amount, versus the baseline, which majority of people, not going to do anything to them. Okay, we're interested in that, but there's no way to do that. There's no way to go about calculating those out in a meaningful way. So it's just not done, even though it's what's interesting or what we need to know. All right, so that brings me to kind of thresholds and what we call the Delaney Clause. Okay, Delaney was a congressman who wanted to put a clause or put an act together that forbidded any additions of any amount of material that's known to be a carcinogen in foods or drugs. Okay? And so this started, I think, in the 60s, the Delaney Clause. It might have been a little bit later in the 70s. And this was the clause. This was passed at first. Basically, if we know it's a carcinogen, it can't go into food and drugs. Well, that's not possible. Okay? So it's impossible to meet that standard, and it was replaced in 1996 by non or no reasonable harm, okay, requirement. So now instead of any amount, it's been replaced with no reasonable harm. Okay, so less than one cancer for every 1 million people exposed over a lifetime. Okay? So the fact that, and I don't have one handy, but the fact that people say, like diet soda causes cancer, right? It can't. Right? It can't. There is no scientific evidence, okay, that suggests that diet soda causes cancer. Now, there are other things that it causes. It can cause weight gain, because eventually the bacteria in your stomach will break down aspartame and use it as a sugar. And so you might at first get this decrease in weight, and then next you get an increase in weight. But it cannot be a carcinogen because, well, first of all, millions of people drink diet soda. And we have this stipulation in 1996 that was put on things. One cancer in every 1 million people exposed over a lifetime. You can't have that. Okay? So if it's a known carcinogen, you can't sell that. You cannot sell diet soda if it was a known carcinogen. Okay? So risk and assessment, again, a little bit about this. Risk is the possibility of suffering harm or loss. Risk assessment is analyzing things, so benefit versus risk kind of situation. A risk assessment, a true risk assessment, if a team's doing this, they're going to first identify what the risk is, okay, and then they're going to identify what's the response, or what kind of response should we have to that risk. Is this a dosage risk assessment? So how much toxicity is there, or pollutants, or things like that? Or is it a risk to bodily harm, okay, running a chainsaw or something like that? And then they're going to say, well, how many exposures are you going to have? Are you going to do this every single day? And then they can classify that risk in certain classification. How risky is this job? And so a lot of people will look up like, oh, what's the most dangerous job? Okay? This is how a risk assessment is done. They look at identifying the risk, what's the dosage if it's chemically based, what's the exposure, okay? And how do we classify that risk? But risk assessments, they're not really rational. They downplay risks to suit their agenda. Okay, so interested parties, let's say, companies that really want people to work for them, they will downplay the risk of being exposure. I mean, how many people who mine for a living know that black lung is a legitimate concern, but their boss will downplay it. No matter what, they're going to downplay how many people are affected by it. Okay? Because they're the interested party. They want you to work for them. They're going to downplay it. Now, are they going to give you safety procedures and other materials and things like that, and have you checked and give you limits on how much time you can spend exposed to things? Absolutely, because they don't want you to sue them, but they're still going to downplay. Okay? We often also tend to tolerate certain risks. Okay? It depends, the risk depends on what we want to do. The risk of, lung cancer for someone that smokes, well, it depends. Do you like to smoke? Right? If you enjoy smoking as something that you like to do, then you're going to tolerate the risk for that. This just depends on lots of things. Individuals that like to drive fast, they get a thrill out of driving vehicles extremely fast. There's a risk behind that. They tolerate that risk, okay? Even though they know that driving speeds of 100 miles per hour or more, if you wreck, the odds are pretty good that you're not going to survive that wreck. I mean, this is often, the risk assessment and the non-rationale based on those risk assessments. The other thing is most people don't understand probability very well. So when I say that 1 out of every 2 people, every 2 males has a potential to get cancer, the probability of you developing cancer is 50%. People don't understand that. They don't understand probability of injury or risk that's involved. Okay? There's also lots of things. I mean, I don't know how many times people have, in live lectures have told me, well, my grandma smoked for 90 years and never developed cancer. Okay, that's a personal experience. Okay? That doesn't mean that you can smoke for 90 years and not develop cancer. It's a probability still. I'm not saying if you smoke, or I'm not saying if you drink alcohol, I'm not saying if you, drive at 100 miles per hour, you're going to wreck, or you're going to die, or you're going to develop cancer. I'm just giving you the probabilities. And that's where risk assessment falls apart, is because people really don't want to look at the probability. Okay? I mean, most people consider themselves above average drivers, but this is not the case. It's not true. Right? You're not an above average driver. Now, some people are above average drivers, right? But most people are actually probably average drivers, and quite a few of those that think they're average, they're probably below average drivers. Right? So often we overestimate things. The media over-reports sensitive things. We have an irrational fear for some things. So for some things, the overestimation is extremely dangerous. I mean, for example, most people are extremely afraid of rattlesnakes. rattlesnakes, you know, they're poisonous, they're venomous. Oh man, they're going to attack you. I'm not going to go to a region. I'm not going to go on a hike where there might be rattlesnakes. Okay? Rattlesnakes in the United States maybe kill one person a year. Maybe. I mean, you know, it's the risk is there, of course. Okay? Black widows. People are deathly afraid of black widows. You know, black widows never killed a human on record. Never. Okay? No one's ever died from a black widow bite. But we over exaggerate the risk that is applied with black widows. Okay? Does it hurt if you get bit? Do you have to go to the hospital? You don't have to. You're going to lose tissue if you don't. But yeah, there's risk involved with that. Okay? But dogs, domestic dogs, the family dog, kill way more children than a black widow, because black widows kill no one. Okay? Way more individuals, okay, than a black widow, but literally everyone accepts dogs into their house. Hardly anyone accepts a black widow into their house. Right. So probability or tolerating probability, it depends on the risk. Okay, so it depends on the individual. You know, great harm is accepted only in very low levels of frequency. So, you know, if you have a chance to say skydive, or you have a chance to bungee jump or something like that, people would normally say, well, the risk is, well, what happens if the equipment fails? Okay, but if you looked at the probability of that happening, it's very, very low. Okay? You know, people have a fear of flying. Right? The probability of an airplane crash, again, very, very low. Right? But because that probability is very, very low of that happening, we accept great harm. You get in an airplane and it crashes, not always, but you're dead. Okay? I mean, that's great harm. But the fact that there's very low of frequency that would ever happen, or if you bungee jump and the line breaks, you're dead, great harm, but very, very low level. So a lot of people will accept that kind of risk. Okay, so for most people, a 1 in 100,000 chance of dying is a threshold for changing behavior. Okay, for some people, it doesn't really matter. Whoops. So if we look at something like the Environmental Protection Agency, the risk that the environmental or the EPA must provide is 1 in 1 million is acceptable for environmental hazards. So I already told you that. Okay, a toxicity level of 1 in 1 million is acceptable. Okay? So 1 in 1 million will develop cancer from this exposure to this chemical. That's an acceptable EPA regulation. Look at this, okay? The risk of dying from lung cancer if you smoke is 1 in every 4 individuals. Okay? The risk of being killed by a shark is 1 in every 264 million people. Okay? Not very many people, when they walk outside and see someone smoking, is afraid of that individual. And that individual clearly is not afraid of smoking. But many people, millions of people, are afraid of sharks. Okay? And if anyone ever said there's a shark in the water, guaranteed you're getting out of the water. Okay? People focus on the latter. Okay? Focus on what they don't know. All right? So when it comes to risk assessment and management, again, like I said before when I started this section, okay, it has to do with your socioeconomic status, so where are you at financially. It has to do with politicals and politics behind it. It has to do with maybe some religion. Okay, it has to do with the hazard that you've identified. So has the hazard even been identified? Okay, The dosage, do you know the dosage? Okay? How much are you being exposed to it? And this will allow you to categorize the risk, right? And take that risk into account, whether you're going to expose yourself to that or not. Right? And some of this stuff, other factors can also be, people chattering in your ear, okay? These kind of things from an individual basis, but it also can be regulatory decisions from EPA and other things. They can be definitely the result of sociology and who's the loudest in the room to get these regulatory decisions applied. Okay. So here, real quick, these are the lifetime chances of dying in the United States. Heart disease is #1, cancer, stroke, motor vehicle accident. I can assure you sharks are not on the list. Floods, dog bites, bees, wasps, stings, lightning strikes. I mean, these are the odds. But, the top 4, heart disease, cancer, stroke, and motorized vehicles. Okay. All right, so establishing health policies, it's difficult. There's so many hazards out there, there's so many risks, and humans tend to avoid risk or, alter those risks based on situations. I mean, I know so many times that, people would never do something, but because their buddies are around or their friends or their family are around, okay, they're going to do it. And they increase that risk because of peer pressure. Most people know situations where this happens. So establishing health policies around risk can be very, very difficult. But we do set standards, and the EPA has standards, and we should always concern ourselves with certain things. First of all, combined effects of exposure. Right? This has got to be key. We have to know what happens when you mix certain chemicals. We have to know what happens when the toxicities are mixed between things, or after it's been broken down, that product. Okay, and we don't have a very good wrap around this. Okay. Different sensitivities. I wish we do this. Okay? Different people are sensitive to different things, and we really should know these kind of things. Okay? And then the difference between chronic and acute exposure. We often don't consider these things. So we have work to do from the Environmental Protection Agency when it comes to toxicity and it comes to the science in developing those regulations. And these 3 things are key to that. Okay. Now, of course, it's not reasonable to say that we're going to be protected from all contaminants. And there is going to be risk in just living. That's clear. But we can minimize those risks as we develop new products, as we develop new medicines, new types of packaging, and things like that, by exposing or doing some scientific research before those products are put out and allowed for the humans that are not expecting to be part of a research project, okay, to be exposed to those materials. And the other thing that we need to do is we need to look at this from an ecotoxicology point of view, and that is we need to really look at what's the exposure, what's the risk to the other organisms in the environment. We're often just concerned about humans, and this has really destroyed a lot of ecosystems and caused a lot of natural resources that we once had to be maybe not extinct, but at least to have decreased populations or be not available as a natural resource any longer. All right, sorry for the super long lecture, but I wanted to finish that topic up so we can move on to some other more pertinent topics to looking at the management of natural resources next time.